DNA replication is the process by which a cell duplicates its genetic material before it divides, ensuring that each daughter cell inherits an identical copy of DNA. In prokaryotes like bacteria, this process occurs at a remarkable speed and precision. Let's dive into how this process works step by step inside prokaryotic cells, focusing on the model organism, E. coli. Replication in prokaryotes begins at a single, well-defined point on the circular chromosome called the origin of replication, or oric. Here, proteins called DNA recognize specific sequences on the DNA and bind to them, causing the DNA strands to unwind slightly. This local unwinding is just the beginning. A protein complex, including helicase, also known as DNAB, is then recruited to continue unwinding the DNA strands, forming what we call the replication fork. As helicase works its way down the DNA, unzipping the double helix, single-strand binding proteins step in to stabilize the unwound DNA, preventing the strands from snapping back together. Now that we have two exposed single strands, we need to prepare them for replication. That's where primase comes in. Primase lays down a short RNA primer, which will act as a starting point for the next phase, DNA synthesis. Section 3, elongation. With the primer in place, DNA polymerase III takes over. It's responsible for synthesizing new DNA by adding nucleotides to the growing strand. The leading strand is synthesized continuously in the 5' to 3' direction. However, the lagging strand presents a unique challenge. Since it's oriented in the opposite direction, DNA polymerase has to work in short bursts, creating segments known as Okazaki fragments. These fragments are later stitched together by an enzyme called DNA ligase to form a complete strand. The Section 4, termination and final steps. As DNA polymerase continues its work, eventually the replication fork reaches a region called the termination site. Here, TUS proteins bind and halt the progress of the replication machinery, signaling that replication is complete. Meanwhile, topoisomerases, like DNA gyrase, manage the DNA structure, ensuring that it doesn't become overly twisted or tangled during replication. Finally, DNA ligase seals any remaining gaps between Okazaki fragments on the lagging strand, producing two identical copies of the genome. And there you have it. DNA replication in prokaryotes is a highly efficient and precise process. This rapid and accurate replication allows bacteria like E. coli to multiply quickly, supporting their ability to thrive and adapt to different environments. Understanding this process is fundamental to our knowledge of genetics and cell biology.